Hello friends, welcome again. Greetings for the day. So in continuation with our previous video, today we are with a new video. So we have started this initiative to help our pharma friends to bridge gap between industry and academia. So today we will cover ICH Guideline Q3B, impurities in new drug products. So let's start. Preamble. So Q3B, it is a step 4 version. The guideline become effective from the 2 June 2006. So this document means ICH Q3B provides guidance for registration application on the content and qualification of impurities in new drug products produced by chemically synthesized new drug substance not previously registered in a region or member state. So in this document we will cover content, that what impurities to be specified in the our specification, how to understand what is the limits for the impurities, and if the impurities is crossing our qualification threshold, then how to qualify those impurities. So all that, identify what is the identification threshold, what is the qualification threshold, all about impurities in drug product. Drug product means finished formulation. So in finished formulation, how to decide the impurity levels, how to decide the limits of those impurities level, that we understand in this guideline. So the guideline address only those impurities in new drug products classified as a degradation product of the drug substance. So the impurities, the unwanted substance, which we call in this guideline as a degradation product. So only degradation products which form by the interaction between the drug substance and excipients. So drug substance impurities we covered in the ICH Q3A guideline. So now in this Q3B we will cover the impurities of the drug product, means finished formulation. These impurity may arise by the degradation products from the drug substance, and how this degradation product will form, they form by the reaction or chemical interaction with the excipients or container closure system in which the drug product is stored. So these impurities, these unwanted products were called degradation product, and only those degradation product which caused by the drug substance by the interaction with the excipient or container closure will be discussed in this Q3B guideline. Okay, so generally the impurities present in the new drug substance need not to be monitored or specified in the new drug product unless they are also degradation product. What it means, that the process impurity means we already have seen the ICH Q3A. So in the organic impurity there are organic impurity, inorganic impurity, residual solvent also. So the organic impurity may come from the process of the drug substance manufacturing, means API manufacturing itself, some process impurity may come. So that process impurity, if it is not the degraded impurity, what degraded impurity with the time being, with the time if that impurity increases, then it is a degraded impurity. If throughout the shelf life of the drug product or drug substance, if that impurity is increasing because of the stress, because of the temperature, because of the exposure to the temperature, because of the exposure to the humidity, because of the exposure to the light, if that impurity is increasing, then it is a degraded impurity. If it is a degraded impurity, then we will discuss here. If it is not, it is a process impurity, we will not discuss here. It is need not to be discussed here, because ICH Q3B focus mainly on the drug product impurity. The process impurity were already controlled by the ICH, by the recommendation provided in the ICH Q3A. So let's start again. So first we understood what is the reporting threshold, what is the identification threshold, what is the qualification threshold, right? These three terms are very important. First is the reporting threshold, identification threshold, qualification threshold. Okay, so which impurity to be reported means the result should be presented for which impurity. That how we decide? We decide by the maximum daily dose, by the maximum dailies of the drug. So based on the maximum daily dose, we provide a limit that we call reporting threshold, that is expressed in the percentage. So if the degradation product, if the impurity is greater than the reporting threshold, the limit provided by the recommendation provided by the ICH based on the MMD, maximum daily dose, so then we should report it. Again, if the degradation product present in the drug product, if the degradation product present in the finished formulation is greater than the reporting threshold, then that impurity should be reported. What is the identification threshold? If the degradation product present in the drug product is greater than the identification threshold, then the structure of that impurity required to be identified, right? What is the qualification threshold? If the degradation product is greater than the qualification threshold, then we either need you either then we need to either reduce that impurity below the qualification threshold, or we have to establish biological safety of that impurity to qualify the observed limit. Which, what it means, if suppose we'll discuss in detail also in the next slides, but just in a brief, if the impurity is coming, the level of the impurity in percent, it is coming more than the qualification threshold. So qualification threshold is determined by the maximum daily dose. Okay, so if the degradation product is coming more than the qualification threshold, then we have two options. Either we have to reduce the impurity below the qualification threshold, or we have to evaluate the biological safety of that impurity to up to the level which is it is observing in our drug product, in our formulation, right? So three thresholds are there. Reporting threshold: if the impurity is above reporting threshold, we should report it. If it is below, we shouldn't report it. Identification threshold: if the impurity is crossing the identification threshold, if it is greater than the identification threshold, then we have to identify structure. If structure is safe, then no issue, that impurity can be present up to qualification threshold. But if structure is not safe, then we have to reduce again below the identification threshold. Right now, we have to reduce that, right? And if the impurity is above the qualification threshold, then we again have to qualify. Either we have to reduce it below the qualification threshold, otherwise we have to qualify the limit which is observing the drug product. Up to that limit, we have to qualify that impurity that it is biologically safe. Analytical procedure and reporting of degradation product. So analytical procedure should be validated to demonstrate specificity for the specified and unspecified degradation product. So whatever specified means known impurity, you know the structure. Unspecified means you don't know the structure. So to check the impurity in the drug product, whatever analytical method to be used, that should be properly validated. Validated means it should be able to distinguish that impurity, each and every impurity. It should be able to distinguish that each and every impurity, and that method should be validated by the ICH with reference to the ICH Q2 guideline. Okay, so the method whatever used to determine the impurity should be validated and should be stability indicating. This is the first thing. Second thing, as appropriate, this validation should include samples stored under relevant stress condition: light, heat, humidity, acid base, hydrolysis, and oxidation. So we know that we do the stress study, we do the force degradation by exposing to the light, heat, humidity, hydrolysis, and oxidation. This is also helpful to know the degradation pathway, that by which pathway the same impurity is generating. So that validation should cover all these stress studies also. Then below, how to report the impurity. See, if the impurity generally reported in the percentage. Okay, so if it is below the 1%, then it should be reported to 2 decimal place, and you can use the general round offing rule for making it to 2 decimal place. So if impurity is less than 1%, report it to the 2 decimal place. If impurity is more than 1%, then report it to the 1 decimal place. For example, if it is 0.067 or 0.057, so it will be reported as 0.06. If it is 1.34, it should be reported as 1.3. One more thing, chromatogram. Chromatogram is we see in the HPLC when the impurity will be let out. Then some chromatogram we get. You can ask your AD colleague; he can show you. So chromatogram with peaks labeled, or equivalent data if other analytical procedures are used from representative batch, including chromatogram from analytical procedure validation studies and from long-term and accelerated stability studies, should be provided. The applicant should ensure that the complete degradation product profile, example chromatogram of individual batches, are available if requested. So whatever batches we are submitting, whatever validation we are providing, the way by which we are analyzing the impurity, we are characterizing the impurity, all are the auditable document, and agency can ask whenever required, right? So this is the analytical procedure reporting of the degradation products. Then our specification. So specification is basically our control strategy based on the development experience, based on the drug substance impurity, based on the drug product impurity, based on the drug product impurity during stability. We make a tabular chart, right? We make a tabular chart. Then we identify, we classify the impurities, and then we make our specification, right? So in specification we check, we provide the limits based on the guidance provided in the ICH guideline. So our drug product should comply our limits in the release stage also, means after manufacturing batch, when it is batch release, then in the release stage also, and throughout the shelf life of the drug product, or in the stability when we determine the shelf life, there also it should meet their specification. So generally specification consists of each specified identified degradation product, means based on the development experience, you know that this impurity is about to come, so it is a specified and identified, it is above the identification limit, so you identified that structure also. So each specified, or you know the structure, that is identified means you know the structure. So each specified means you know that it will come, identified structure is known, degradation product. Each specified unidentified degradation product, each specified you know it will come, but unidentified means a structure is not identified because it is below the identification threshold. If above the reporting threshold you will report it, below the identification threshold you will not report it, right? So it will be the below the, it is below the identification threshold, so you're not identified, but you know that impurity will come. So each specified unidentified degradation product, any unspecified means unknown degradation product, right? Now within acceptance criteria of less than equal to the identification threshold. So any unknown impurity, unspecified degradation product, that value is less than equal to the identification threshold, that's why you didn't identify that structure, okay? And total degradation product means some summation of all the impurity which is observed, that is the total degradation product. So these are the elements of the specification, these are the elements of our control strategy which should be inbuilt in our specification to ensure that drug product have impurities degradation product within limit. So regarding thresholds, so uh, we already discussed that three thresholds are there: reporting thresholds, identification threshold, qualification threshold. So it is an important part. So we will give some time here so that we become through with this. So reporting threshold. If impurity is more than reporting threshold, then only we have to report it in the analytical reports. Okay, in the results. Okay, so if it is more than reporting threshold, then only we have to report. Then identification threshold, if it is more than the identification threshold, then only we have to identify its structure. Then qualification threshold, if it is more than qualification threshold, then either we reduce it to below the qualification threshold, or we need to qualify that impurity. We what qualify means, we need to ensure biological safety of that impurity up to the level, up to that which level it is observing in the drug product during shelf life or stability study, basically during the shelf life, right? So these are the three thresholds. So how these threshold determine, these threshold are provided in the these threshold to determine the value of these threshold, the recommendation is provided in the ICH guideline. Okay, so these threshold determined by the maximum daily dose. Okay, so reporting threshold, for reporting threshold, we, if the maximum daily dose is less than equal to 1 gram, then reporting threshold will be 0.1%. Up to what a level the impurity should be reported based on the threshold level provided in the guideline. So less than 1 gram, the threshold will be 0.1%. Greater than 1 gram, reporting threshold will be 0.05%. Okay. For identification threshold, MMD is divided in the MMD, maximum daily dose, is divided as less than 1 mg, 1% or 5 microgram TDI, total daily intake. 1 mg to 10 mg, if maximum daily dose is, then threshold will be, identification threshold will be 0.5% or 20 microgram TDI. If the maximum daily dose is greater than 10 mg and up to 2 gram, then 0.2% or 2 mg TDI will be the identification threshold, and if it is greater than 2 gram, then 0.10% will be the identification threshold. For qualification threshold, less than 10 mg, 1% or 50 microgram TDI, 10 mg to 100 mg, 0.5% or 200 microgram TDI, greater than 100 mg to 2 gram, 0.2% or 3 mg TDI, greater than 2 gram, 0.15%, is the threshold. So let understood by an example, so it will be much easier. So suppose if a drug having 50 mg per day maximum daily dose. Okay, so a drug product have a 50 mg per day maximum daily dose. These are the raw data which obtained by the analysis. So first we need to understand whether these data to be reported or not. So 50 mg per day will fall in this category, because the 50 mg per day will fall in the this category, that maximum daily dose is less than 1 gram, right? So the threshold will be 0.1%. So here threshold will be 0.1%. 0.04 is less than the 0.1%, so this will be not reported. Zero point 0.2 is more than the 0.1%, so it will be reported as a 0.2. Then 0.349 means if we round off, then 0.35, then also 0.3 only, so it will be reported it is more than the 0.1%, reported as 0.3. 0.55, if we round out, when it is 0.6, it is more than the 0.1, so it is reported as a 0.6. So these three impurity will be reported. This impurity will be not reported. Why it is not reported? Because 0.04 is less than the 0.1%. Again we will repeat, 50 mg mmd, so fall in this less than 1 gram category. Threshold will be 0.1%. So above 0.1% only reported, all impurity only reported. 0.04 not reported. 0.2 yes reported. 0.3 yes reported. 0.6 yes reported. So these three impurities is reported. So by this reporting threshold we can calculate the TDI. See here it is written, for identification threshold I am talking. So 1% of 5 microgram TDI, you have to calculate the TDI base either 1%. So based on the TDI, total daily intake, TDI means total daily intake. If we calculate 1%, so if it is 1% is lesser, we should consider that, or if 5 microgram is lesser, we should consider that. So this should be considered from here only. Okay, so for example, if 50 microgram 50 mg per day is the maximum daily dose and 0.04 is the impurity, then how much impurity, total degradation product, how much total daily intake of the degradation product is how much? How to calculate? See, 50 mg is the maximum daily dose. How much this impurity is coming? 0.04. So how much it is coming? 0.02 multiply by 1000, so it will become around 20 microgram. Okay, then the impurity is coming 0.2%. Again 50 mg is the daily dose, maximum daily dose. 50 mg multiply by 0.2%, it will come 0.1 multiply by 1000 to convert in the microgram, it will come 100 microgram, right? So it is 100. In similar way, 0.3 multiply by 50 percent, 0.15 multiply by 1000, 150. In similar way, 0.6 multiply by 0.5, 0.6 multiply by 0.6% multiply by 50 mg, and then multiply by 1000, it will come around 300 microgram. So these are the total daily intake of the individual degradation product which is observed here. Okay, so let's check the identification threshold. So 50 mg, it will fall in the category greater than 10 mg, 2 gram, right? So what is 0.2%? What is the limit for the identification threshold? 0.2% or 2 mg, whichever is lower. So 0.2% or 2 mg. So for this impurity, 0.2% is coming around 100 microgram, right? For this impurity, it is coming around 150 microgram. For this impurity, it is coming out 300 microgram. So all 100, this will be not reported, just we calculated for reference purpose only. So for all this, it is coming 0.2% is coming very less than the 2 mg TDI. So our limit will be, our identification limit will be 0.2%. Okay, so our identification limit will be 0.2% only. So if we see from the 0.2%, which impurity is increasing? It is increasing, no. It is increasing, yes. It is increasing, yes. So these two impurity are more than the identification threshold, so their structure to be identified, right? Then comes for the qualification threshold. So how this qualification threshold 200 microgram came? So again we'll go through the table. So it is a 50 mg dose. This is the qualification threshold table. So it will fall in the second category, 10 mg to 100 mg. So 0.5% should be the qualification threshold. Let's we calculate 0.5% for the for this impurity only. We calculate this impurity. So it is coming around 0.3. Okay. So 50 mg multiply by 0.50 mg. So how we calculate? This is the maximum daily dose, 50 mg. So 50 mg multiply by our qualification threshold means 0.5%. So if we do the 0.5%, it will come around 0.25. So 0.25, if we multiply by 1000 to convert it into microgram, it will come around 250 microgram. So 0.5% limit for 50 mg per day comes around 250 microgram. So it is more than the 200 microgram TDI. So that's why 0.5% limit will be not followed, 200 microgram limit will be followed. So again, how the qualification threshold is 200 microgram, how to calculate? So 50 mg, it will fall in this category, 10 mg to 100 mg for qualification threshold. Okay, so in this category, either 0.5% should be a limit or 200 microgram TDI, total daily intake, should be a limit. So how to calculate? 50 mg, whatever your maximum daily use, multiply by 0.5%, right? It will come 0.25, multiply by 1000 to convert it in the microgram, because the impurity, the MMD is provided in the 50 mg, so it will come around the 250 microgram. So 250 microgram is more than the 200 microgram. Whichever is lower, that limit you have to consider. So that's why we will consider qualification threshold 200 microgram limit. So if we see the microgram intake of the derivation product, this only, this third last one is crossing the 200 threshold. So this impurity need to be qualified. These two impurity need to be identified, and this last impurity need to be qualified. Hope you are clear with that. If you have any issue, then you can give a comment so that we can arrange a live session for that also. So we will check one more example. So here the MMD is 1.9 gram per day. Okay. So here MMD is 1.9 gram per day. So we will again, these are the raw results, 0.049, 0.0. So if 1.9 gram per day, it will fall in this category. So limit will be 0.05 for the reporting threshold. So if we see the round off in up to the 2 decimal digit, 0.05, it will be not reported, because 0.05 after round offing, 0.05 is equal to 0.05, so it will be not reported. 0.08 is bigger, is more than the 0.05, so it will be reported. 0.18 is more than the 0.05, it will be reported. 0.19 is more than the 0.05, so it will be reported, right? Then we consider that, then we calculate the total daily intake. So 1.9 gram means it will be fall in this category only. So either 0.2% or 2 mg will be the identification threshold. So this we will discuss later. First, how this value TDI is calculated, TDI of the degradation product. So MMD multiplied by the impurity observed. For example, 1.9 is the MMD, multiplied by 0.05% right now. So if you change in the micro in the mg, because it is gram per day, you have to multiply by 1000, so it will come 0.95. If you round off it to 1 mg, so 1 mg will be the TDI for the first impurity. For second, 1.9 multiply by 0.08%, okay, and again multiply by 1000, it will come around 1.52. So if you round off, it will come around 2, okay. Then 1.9 multiplied by 0.18%, so it will come around 3.42. So if you round off, it will come 3, okay. Then last one, 1.9 multiplied by 0.19%, it will come around 0.0036, 3.61, round off, it will come around 4. So this is the mg total daily intake of each degradation product. Then come to the identification threshold. How the identification threshold will be decided? So 1.9 gram is the MMD. It will fall in this category. Let's calculate 1.9 multiplied by 0.2%, right? So it will come like that. If you multiply by 1000, it will come 3.8 mg, means 4 mg. So 4 mg is much larger than the 2 mg. So 2 mg TDI will be the identification threshold. Same we can check the qualification threshold. For qualification threshold also, the maximum value is 1.9 gram, means it will fall in this category. So again 1.9 multiplied by 0.2% into 1000 to change in the mg, so it will 3 point means 4 mg. So 4 mg is also more than the 3 mg, so 3 mg. So this way, this identification threshold 2 mg is decided, and qualification threshold 3 mg is decided. Then we will check which impurity is increasing. So this is increasing, no. This is increasing, no. So this impurity 3 mg increasing, yes. This no. This increasing, yes, yes. So these two impurity you need to identify structure. For this impurity you need not to qualify. You can use the qualification threshold as a limit in the specifications, but for this impurity you need to qualify this impurity. You need to establish the biological safety of this impurity. Okay. So now whatever we learned, we will discuss in the decision tree provided in the ICH guideline. So first we have to check whether the results which we obtain is more than the reporting threshold. If it is more than the reporting threshold, then only we have to report those impurities, those degradation products. Okay. So after reporting them, if the degradation product value, whatever value we are obtaining, it is more than the identification threshold, then only we have to go for the structure identification. Okay, if it is less than the identification threshold, no action required. If it is more than the identification threshold, then we have to identify its structure. If its structure is toxic to the human, then we have to reduce it below the safe level, means below the identification threshold. So if structure is identified, then we have to check whether any human relevant risk is there. If yes, we have to reduce it to the safe level. If no risk is there, then we have to check whether the limit is crossing the qualification threshold. If no, if it is below the qualification threshold, no issue. If it is above the qualification threshold, so we have to either reduce it to the qualification threshold, or we have to go for the we have to go for establishing the biological safety. So biological safety, in which we check for, basically we check for the genotoxicity study. So if in the genotoxic study, what we do to make that level biological safe, to establish that level biological safe, if no adverse effect, then we can qualify that limit, whatever we are observing or whatever limit we have studied, that we can qualify for that particular impurity. If some adverse event is observed, then we have to reduce that impurity to the safe levels. Okay, so this is all about the ICH. This is all about the ICH Q3 guideline. Thanks a lot. Thanks for your time. So if you enjoyed the video, if you got some learning, then like this video, subscribe the channel, share it wherever possible, so that more people get benefited from this, and we also get motivation to bring this type of more videos, more learning videos. Thanks a lot. Thank you. Bye bye.